Yes, madam.
Okay. Good afternoon to everyone. And is my voice clear so that everyone can hear me clearly?
Yes, ma'am.
Yes.
Okay. Thank you. This lecture. It's about introduction to some of the biosensing techniques, and I want the session to be interactive. So if you have any questions, you can ask me then and there. And I would like to thank Alvin and Department of Biomedical Engineering for giving me this opportunity. This is the session contents where I'll be briefing about the different biosensor techniques which is being employed, and which includes fiber optics, surface plasma resonance, interferometric biosensors, and SCRS. These are the major categories. Before going into the details, I'll explain you what a biosensor is. Has anyone familiar with the terminology biosensor? I think you in this course you have already attended 2 lectures, right? Can anyone explain me what a biosensor is? Yes? At least can you give some examples of biosensor? Can I call a thermometer as a biosensor? Any response? I hope I am audible to everyone, right?
Yes, yes, yes.
Yes, yes, yes.
I think they are very stubborn not to open their mouth. Okay, fine. A thermometer, we cannot call a thermometer as a biosensor because it's a biomedical sensor where it is employed only for measuring the physiological parameters. So, what is the difference between a biomedical sensor and biosensor, and why it's so unique for a biosensor? I'll play a small clipping where you could understand what a biosensor is. A biosensor is nothing but a sensor. You detect some parameters of interest with the help of another biomolecules of interest. These are called biomarkers. These biomarkers present in the blood sample, urine, sweat, whatever. So it is present in the biosample, and if you look at here, if you take the blood sample where the plasma contains these biomarkers of interest, and when you introduce the blood sample to the device, monitoring device, it will display you the concentration of glucose or concentration of any other protein markers or concentration of any other metabolites. So basically it is a molecular recognition with the help of another molecule. These are the bioreceptors, antibodies, aptamers, enzymes. These are some of the bioreceptors which can be utilized for detecting analytes of interest. And you can use different transduction techniques. Maybe I'll come to the transduction technique a bit later. So with this biosensor, we could develop many applications. It's not just clinical diagnosis, it is also employed in various other fields. In clinical diagnosis, we identify pathogens, virus, metabolites, etc. And we also use it for food quality application, where the quality of food will be monitored with the help of this biosensor, like the taste, sweat, and other things. This is one example where you analyze the exhaled breath and try to identify the pathogens present. This is also a kind of biosensor which is being employed. And the biosensor is also used in environmental monitoring, the soil moisture content, humidity, and biotoxins present, and some other environmental gases. So biosensing, it's not just restricted only to clinical diagnosis, it is employed in various other fields like environmental monitoring, food quality control, and in automobiles, and in concrete structure design, and then the strength evaluation. There are many other applications also. The key is you have to have a bioreceptor to identify the biomolecule of interest. This is the block diagram or the entire components involved in developing a biosensor. If you look at here, the analyte which is present in the biosample, which is nothing but glucose in a blood, is an example, classic example which I use for explaining this concept. And it has to be interacted with the bioreceptors. The bioreceptors again can be categorized into different categories: antibody, enzymes, cells, aptamers. These are nothing but a biomolecule. With this biomolecule I am going to detect the analyte. Analyte is also another biomolecule of interest. So the only difference between a biosensor and biomedical sensor and other physical chemical sensor is you need a bioreceptor or a live moiety for detecting the analytes of interest. So when you coat these bioreceptors onto a transducers, the transducer, you can have different substrates like plastic or metal or paper. If you look at pregnancy test strips, they use nitrocellulose membrane paper as a substrate, and if you look at glucometers, where they use a plastic strip as a substrate. So once these bioreceptors are contoured onto these substrates, you can convert the signal transduction. What is the signal transduction here? A biochemical reaction between the bioreceptor and analyte. That biochemical reaction, we are transducing it into different, using in different categories listed over here. You can have an electrochemical biosensor where you will be interested in measuring current or voltage generated equivalent to the biochemical reaction happening, and optical biosensor where you will monitor the changes or the presence of analyte by means of changes in the properties of light signal, and thermal where the temperature changes, and electric again it comes under electrochemical, acoustic. So there are different transducers which could be employed to read this biochemical reaction, which will give us information about the biomarkers of interest. Biomarkers are nothing but which cause the disease, and then it can be interfaced with any of the real time devices like a smartphone, laptop, PC, etc. So for detecting these disease causing analytes or the biomolecules which cause diseases which is present in the biosample, the major biosample which is currently we are utilizing in clinical laboratories are blood. So some other biosamples, if you want to explore: urine, saliva, sweat, tears. These are some other biosamples which are being explored to detect the disease causing biomarkers of interest. The concentration of these biomarkers will be very less in the biosample, and that's why there is a huge demand lies on the development of these biosensors to improve the sensitivity or to enhance the detection limits. If we look at the clinical laboratory setup, if we give the blood, first they will centrifuge and they separate into cellular and the plasma component. The biomarkers which I am talking, it is present in the plasma component, and based on the type of analyte and the type of bioreceptor utilizing, the assays are classified into different categories. One is immunoassay, where an antigen and antibody interacting, the biochemical reaction between the antigen and antibody will be monitored or will be realized. And another one is aptasensors or nucleic acid sensors, where the interaction between the nucleic acids resulting in hybridization of these nucleic acid sequences will be exploited to identify the biomarkers of interest. Another one is peptide, which is a amino acid small chain, and that is also used for detecting proteins. And another one is lectin protein interaction, where you have a protein which is sensitive to glycogen and lipids. That is called lectin protein combination or lectin protein assay, where the lectins are utilized for identifying the proteins present in the biosample. And another one is the catalytic biosensor, which is based on enzymes. If we look at the commercial glucometers available, they work on the principle of these catalytic biosensors, where a glucose oxidase enzyme is coated into the test strip where we are inserting a drop of blood, and when it interacts with the blood, glucose interacts with the enzyme and generates H2O2 hydrogen peroxide. It's a biochemical reaction equivalent to the concentration of H2O2 generated, the equivalent current signal will be generated, and that we have converted into 120 mg per deciliter or 80 mg per deciliter. So we convert that into equivalent concentration of glucose. This is an indirect detection, but still it's a classic example of enzyme based biosensor. Even pregnancy test strips which has been commercially available, they are also detecting the presence of beta hcg hormone by utilizing the immunoassay, where they are using antibodies for detecting the beta hcg hormone. So it's an example of immunoassay. These are some of the receptor categories and the type of biochemical reaction which could be made possible to detect these analytes of interest. I was discussing about different reactions, right? So catalytic reaction, where enzyme will be coated onto the substrate when the analyte comes and binds. When glucose from the blood sample binds with the enzyme coated onto the test strip, it undergoes a catalytic reaction and produces equivalent electrochemical change or light change, depends on the type of transduction which you are employing. And this is an immunoassay where you have an antibody coated onto the substrate when the antigen comes and binds. Antigen is nothing but the analyte or biomarker, disease causing agent, comes and binds, and they have a specific affinity. This is called affinity reaction between the antigen and antibody, and this is realized in all the immunoassays. So pregnancy test strips, they employ this immunoassay. ELISA, that enzyme linked immunosorbent assay for detecting HIV virus, also employs immunoassay. And this is one another type of study where nucleic acids are employed. You employ a single strand DNA sequence and the analyte, if it is a genetic molecule gene, or if it is any nucleic acid, then it will come and bind, causing the hybridization reaction onto the substrate surface. Then we'll be monitoring this. And another one is aptamers, which is equivalent to nucleic acids. These are commercially or artificially synthesized nucleic acid sequences. In case of nucleic acid, you could detect only the DNA or RNA sequence by employing another DNA sequence or RNA sequence. Whereas in case of aptamers, you could employ this commercially synthesized nucleic acid sequence, and this could be utilized for detecting a peptide or detecting a protein or for detecting another nucleic acid sequence. That is the beauty of aptamers. This could be commercially synthesized, and according to the pathogen, type of pathogens which you have, bacteria or viri or any other metabolite, you can synthesize the sequences. The nucleic acid bases could be generated and further employed for detecting the peptides or proteins or any other small molecules also. Out of all these reactions, this immunoreaction and enzyme reactions are robust compared to the immunoreaction, and nucleic acid sequences, the aptamer based assays, are also robust. Whereas in case of immuno, you do require some experimental conditions to be maintained, like temperature, pH, for the protein to be alive. So compared to all these things, immunoassays are quite sensitive technique. And these are biomarkers. As I mentioned already, different diseases are caused by because of the presence of certain biomarkers which are overexpressed in our human body. For example, in case of cardiovascular disease, troponin is a protein which is overexpressed, and breast cancer, BRCA gene, so that gene is overexpressed in the blood. The objective of the biosensor is to detect the concentration of these BRCA gene or the troponin protein when it is available at a very low concentration, so that you could achieve or you could provide early diagnosis. Again, biomarkers are categorized into different categories. DNA based biomarker, as I mentioned already, BRCA gene, it is a DNA based biomarker. Troponin for cardiovascular is a protein based biomarker, and also we have lipid and sugar based biomarkers also. So all these biomarkers, they are mainly utilized for screening the disease at an early stage, and to identify the prognosis of the disease, how the disease is being progressed, so that you could understand the pathophysiology better. That is the ultimate aim of these biomarkers. And as I mentioned, we have to code these receptors, and there are different techniques to code, or we call it as an immobilization, to immobilize these bioreceptors onto the test strip or onto the nitrocellulose membrane, either by physisorption or entrapment or by means of some chemisorption, where surface chemistry and few functionalization protocols are involved to hold these bioreceptors active and alive without losing its inherent properties. So the main key here is these bioreceptors have to be entrapped onto the substrate, and the activity has to be retained, only then the analyte could be detected, and also the density should be higher. For that we use some surface functionalization chemistry to improve the bioreceptor density onto the substrates. Now, up to this, I tried to give you an idea of what a biosensor is, what are the different building blocks involved in it, and now we'll see some types of biosensors. So, first is the fiber optic biosensor. Are you familiar with optical fibers? Do you know what is an optical fiber and where do we employ optical fiber? Yes?
In wireless
In
data
wireless
transfer.
data transfer. In wireless data transfer, in 5G communication, whatever the communication which we are talking today, 4G, 5G, and the broadbands which we are talking today. So fiber optics it's used in communications, but I am saying here it could be utilized as a biosensor. Can you try to correlate how? What is an optical fiber basically?
It, uh,
It,
it,
uh,
it,
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 uh,
it, uh,
it
it
utilizes total
utilizes total
internal
internal
reflection
reflection
for the
Yes.
uh, for the
For the
optical
optical. Basically, it's a light waveguide. It will carry light from one place to another place, right? So basically, it's a cylindrical tube which carries light from one place to another place. So if based on this application, you could see it is used in interior decorations, and also in automotive industries. Some of the sensors were employed with the help of this optical fiber. In endoscopes, medical endoscopes, where we want to see or visualize the internal organs. So I'll send a light and I'll collect the light back with the help of the optical fiber. And it is also used in TV cables, and as he correctly pointed out, it's used in communication. On the right side you could see an image how light is being coupled or carried with an optical fiber. It's based on TIR, and you know the physics behind. Based on the refractive index contrast of the core and cladding, the light is taken or light is carried with lesser loss or lower loss. That's why it's employed in communications compared to the electrical cables, because of the lower loss it's being generated while communicating the data. And also we have different categories depending on the type of material which the fibers are fabricated, and also depending on the type of light propagation, the way with which the light can be traveled. If you could see, it's a single mode fiber where only one path of light, where the light takes only one single path for it to transmit from one end to the another. In case of multimode, it takes many different paths to reach from the input side to the output side. So this is a bit of physics where the light is being carried from one place to another based on the refractive index contrast of the core and clad, and depending on the type of fiber you will have the different light intensity at the other end. How it's going to be employed for biosensing application? If you look here, basically this fiber optic biosensor we categorize into two categories. One is extrinsic, another one is intrinsic. In case of extrinsic, where I am utilizing the fiber for just carrying light, which is its application, right, which we are familiar with, and that's how it's employed in communication. So similarly here I am going to send light with the help of an optical fiber. I'll carry it and irradiate or inject it onto a substrate, maybe the test strip or the pregnancy test kit, that nitrocellulose membrane, where I'll irradiate light, and I'll collect the light scattered from these substrates, and then I monitor it at the other end. This is called intrinsic, where I'm just utilizing the fiber as a light guide alone. Another category is called intrinsic fiber sensor, where I alter the geometry of the optical fiber so that I could detect the changes in the light at the other end. So basically for the fiber optic sensor we need a light source and we need a photodetector. And when light is coupled or when it is transmitted within the optical fiber, if we change the geometry or if we do any modifications to the fiber, that will be reflected at the other end. That is how we are going to employ it for detecting biomolecules of interest. This is called evanescent wave absorbance. You could see in the figure, generally the optical fiber consists of a core of refractive index N1 and a clad of refractive index N2. What I have shown here in the figure, that grey shaded portion, I couldn't point it out. I hope you can see the image. The grey shaded portion where I have removed the cladding, that's called decladding, so that at the core clad interface you have a exponentially decaying electric field present at the core clad interface. So the below figure, the blue color figure, you can see a mode of the optical light which is transmitted inside the core, and this optic light mode at the core clad interface you could see an exponentially decaying wave, that is called as evanescence. At the core clad interface we have this evanescence present, and when I keep my blood sample or any biosample where I want to detect the analytes of interest, and when I irradiate with light, the presence of these biomolecules will be detected by monitoring or measuring the properties of the light at the other end with the help of a detector. This is called evanescent wave absorbance, and DP is a terminology. I hope you could see the marking of DP in the picture. That is called depth of penetration of these evanescence at the core clad interface. To what extent I can feel the presence of this evanescent field. So if that is defined by the incident light wavelength, angle of incidence, and then N2 and N1. So basically the N2 and N1 of the optical fiber decides this depth. Accordingly you could detect the biomolecules of interest. Is that clear? Where I remove the clad and replace the clad with a biosample, that will act as a clad, and due to the light interaction I could detect the analytes of interest by monitoring the uh, optical intensity or phase or wavelength changes. So it's an example of intrinsic sensor. You could see in this figure where how these fiber optics geometry or the shape of the optical fiber can be modified by means of some mechanical means or by applying some laser irradiation or applying from some force. So in the first figure you could see a coiled fiber, and in the second figure where we have created a hole in the optical fiber, and that could be employed, and that hole and the sample within that hole can act as a cladding, and then the core of the optical fiber, due to the refractive index contrast, it could be utilized for sensing. A U-bend probe, and it's a tapered probe, double tapered probe, and below you could see a corrugated structures and D-shaped fiber structure. So in case of this intrinsic fiber sensor, the geometry of the optical fiber can be modified. So here we classify the optical fiber sensor category to be based on monitoring the light properties. So what are the properties of light? What are all properties we can list for a light? It's an electromagnetic wave. If I shine light, what will happen? If I shine light from one end to another, say for example, or let us take the classic example of sunlight. Sunlight, it falls on our earth. What do we observe from the sunlight? The sunlight could be absorbed, it could be scattered, it could be reflected, and we do see each other because of the scattering phenomena, because of the light which gets reflected in all 360 degrees, so that we could visualize the objects. So there are different properties like absorbance, transmittance, reflection, scattering, refraction. By monitoring these properties alone, I could detect the biomolecules of interest, where I employ this configuration, remove the cladding, and at the photodetector end, so one side I have the source, other side I have the photodetector, and in this figure you could see here there is a plot of wavelength versus absorbance when the pink colored circles represent nanoparticles. I'll come to the nanoparticles bit later. Basically we use these nanoparticles for sensitivity enhancement, and when the analyte comes and binds with the nanoparticles, I observe an increase in the absorbance, as that is delta A, changes in the absorbance intensity, as well as delta lambda, changes in the wavelength. That means absorbance and wavelength properties will be changing. Just a minute, hold on. Yes. Can you see the screen?
Yes.
Yes. So in intrinsic fiber sensor, one category where I am changing the geometry and monitoring the properties of the light. Okay. Another category is where I can employ different substrates. It's a optical waveguides could be employed. Can you give an example of an optical waveguide? Up to this, is that clear? What is a biosensor? And in fiber optics, we are employing optical fiber for detecting the biomolecules of interest. So you can consider the classic example of a glucose sensor, which is a biosensor, where the substrate will be coated with the enzyme. Analyte is the glucose molecule, and when the glucose interacts with the enzyme, it produces some changes in the properties of light, and I measure the concentration of glucose by monitoring the changes in the properties of light. It could be intensity, phase, wavelength, whatever it could be. Okay. So another category, as I mentioned, substrates, optical wave. For realizing fiber optic sensor, you need an optical waveguides. That means any substrate which allows light to travel through. So one such classic example is an optical fiber. Another, you can take planar waveguides. Even the glass lights, you can take the transparent glass lights. It's because it's transparent, so it allows light to be traveled through. So any planar waveguides or the waveguides which allow light to travel through can be used as a substrate. And another category is where I employ these substrates, where I monitor the proper changes in the properties of the light. I want to improve my detection limits. That means I want to detect one glucose molecule present in a blood sample which consists of 10,000 molecules. So detecting one molecule among the 10,000 molecule requires more accuracy and sensitivity, right? So for sensitivity improvement or sensitivity enhancement, we take the help of nanoparticles. I'll come to that bit later. Can you just explain what is a nanoparticle, or have you heard of it before? What a nanoparticle is?
It's a particle which has an order of space of nanometers.
So, what is the order of nanometer means? Can I call 1000 nanometer as nanoparticle?
Uh, 1 nanometer, 110 power minus 12 meters.
1 nanometers to 100 nanometers. If the particles or molecules are below less than 100 nanometer, we call it as a nanoparticle. So, when the bulk metal or material is brought down to the nano regime, they actually behave differently. So what we are going to do here, we are going to exploit the different behavior for detecting this one among the 10,000, so that my sensitivity could be improved. Okay, so I'll come to this bit later. Up to this, is that clear? Are you able to follow? Yes. Are you able to follow, or I
Yes.
hope you had two lectures also in line with it, so I hope you could understand whatever I'm talking. Shall I continue further?
Yes, ma'am.
Yes, thanks. So this is another category of fiber optic sensor where a fiber Bragg grating is employed as a sensor. So can you give an example of a sensor to measure temperature? So which sensor we can employ to measure temperature?
Thermometer or TDs.
Thermometer or RTDs like thermistors or thermocouples could be employed. To monitor pressure, strain
Barometric sensor.
gauges. Yeah, so some sensor we are employing here to measure these physical parameters. So similarly here, the fiber optic, that optical fiber cable which we are using in communication, and it's a long cable, we are going to utilize that long cable as a sensor here. So here I have showed you different geometries where this different geometry optical fiber probe is a sensor, and on top of these substrates surface I coated with the bioreceptors, and I introduce the blood sample so that it will detect the biomolecules of interest. So that is how the fiber optic sensor works. You have the particular geometry, remove the cladding, and instead of cladding you coat with bioreceptors and introduce the analyte. So it's coiled, tapered, U-shaped, all these things work like that. So here it is another example where we employ fiber Bragg gratings. Grating is nothing but periodic lines. So what do we do? Already we know core as a refractive index, right? And the cladding has a another particular refractive. That means core and clad will be of different refractive index. That's why we represent it as N1 and N2. In case of grating, what do? We, with the help of laser inscription, we take a silica optical fiber, and with the help of laser inscription we inscribe lines. So in the left side of the figure, top left side of the figure, you could see a core with some blue equally spaced lines inscribed onto the core of the optical fiber. These are called grating structures. So these grating structures are inscribed onto the core of the optical fiber with the help of a laser inscription, and they are equally spaced. So the interval between the grating lines created, they are equally spaced, and that's represented by the period inverted cap. Okay, and when we couple light through this fiber Bragg grating, so you could see a core refractive index. Initially it was constant, and in the grating structure we have a step index kind of profile because of the periodically spaced laser inscriptions. And through this structure, when I couple light as in you send light from one end here, and at the other end you keep a photodetector and monitor the light coming out through this grating structure. So this is the input where I send a white LED or any light source spectrum. And if you look at the transmitted spectrum, so below figure, on the left side below figure, you could see a transmitted spectrum and reflected spectrum. A particular wavelength of light will be reflected because of the inscribed grating structures, and this could be utilized as a sensor. How? Where I introduce analyte, and I particularly monitor the intensity of the reflected wavelength. If there is any changes, if I vary the concentration of glucose in the biosample. So that is one way where this Bragg ratings are employed for monitoring the biomolecules of interest by simply monitoring one particular reflected wavelength. Okay, and in this Bragg ratings we have different categories: long period grating, tilted grating. On the right side, if you look at, we have a light and spectrum, and as I mentioned already, if light is passing through the grating structure, a particular wavelength of light will be reflected. So in the transmitted signal you have one particular wavelength which is being reflected, and the wavelength which is reflected, that is lambda b, it's dependent on 2n effective and inverted cap period. So it is dependent on the refractive index contrast of the core and cladding, that is defined by n effective, and another one is the period. Period is nothing but the equally spaced grating structures. So the distance between the subsequent or the consecutive grating structures is called the period. So dependent on which you have a particular wavelength. So you could play with the reflected wavelength by fine tuning the core and clad refractive index and then the grating intervals. In case of long period grating, if you look at, they are spaced at a longer distances. Each grating interval is quite long, and the reflected wavelengths, you have multiple reflected wavelengths possible, and those are sensitive to the refractive index changes. SRI means sensitive to the refractive index changes of the cladding structure. So in case of long period grating, it's defined n core minus n clad multiplied with the period of the grating structure. And in case of tilted fiber grating, we have a tilted fiber grating figure below at the end of the right side. When you have a grating structure tilted at a particular angle, so when you are inscribing with a laser and you are making this line, you make that line at inclined at a particular angle. So there will be multiple reflected wavelengths possible, one, and another thing is you can play with whatever the multiple wavelengths, it's dependent on the angle of inclination theta. So this could be employed for multiplex detection also, where in a blood sample, if I want to detect a glucose and then the protein, then I could utilize this tilted fiber gratings, and I could monitor multiple reflected wavelengths and the changes in the multiple reflected wavelengths for the changes in the concentration of the analyte. So this lambda of the tilted fiber grating is dependent on n effective minus n core into inverted cap by cos theta, where theta is the angle of inclination of the grating structure. So this is another category of fiber optic sensor where a Bragg grating is employed for detecting biomolecules are present in the blood sample. And next category is interferometers. Have you studied interferometry before in your 12th standard physics? What is an interferometer and what is the principle of interference? We send two beams and we allow it to travel through or propagate through a certain distance, and you monitor after a certain distance what happens to the two beams, whether they interact with each other, and after they interact with each other they form a certain diffraction gratings. Do you remember that physics? So here also what we are going to do, by employing some optomechanical components, we are going to develop a interferometry setup, and from the reflected light from the sensing region we are going to utilize or exploit that reflected wavelength for detecting the biomolecules of interest. So it's basically we want to measure the analytes by measuring the small changes produced in the path length of the interferometry. So pi is nothing but the phase change which is being generated is dependent on 2 pi ln, where L corresponds to the path length the beam has travelled through. Okay, for example, if I consider in the Michelson interferometer setup, I have placed two mirrors, right? So the distance between the beam splitter, it is represented as BS here. So the distance between the beam splitter and then the mirror M, which corresponds to the path length L, the path length taken by the light to travel through, and N corresponds to the refractive index of the medium, and lambda corresponds to the incident light wavelength. and due to which there is a change in the phase. So in case of fiber Bragg gratings, there will be changes in particular wavelength we are interested in measuring, and in case of this fiber optic different geometry and evanescent wave, we are interested in measuring the intensity. So this is an intensity modulation technique, wavelength modulation technique, and this is the phase modulation technique, where I am measuring particular property of the light for measuring the biomolecules of interest. So in interferometry we have different categories depending on the experimental setup utilized. In case of Michelson interferometer, we need two mirrors and a beam splitter. If you look at from the left side, a light is being incident onto the beam splitter, and that's split into two. One falls onto the mirror down and one falls onto the mirror kept horizontally, and we collected reflected light from both the mirrors. So the phase change or the phase difference generated or created between the two beams will give you information about the biomolecules of interest. This is in case of Michelson. In case of Maxender, where they have utilized two beam splitters and two mirrors. So in the different types of interferometric setup, the only difference is the difference in the alignment and then the usage of the optical components, optomechanical components for allowing the light to travel through. And in case of Fabry-Perot cavity, it's like a resonant cavity where we have kept two mirrors horizontally placed, and the light is being allowed to take multiple paths between the mirrors, so it acts as a resonant cavity. And due to the resonant cavity created between these two mirrors, there will be a phase change, and we are measuring the phase change at the other end of the optical setup. This is about interferometric biosensors, where we are utilizing the property of interference between the light beams by utilizing these optomechanical components. So if you look at here, we are measuring the changes in the path length and the refractive index contrast, and also the wavelength, due to which there will be a phase change phi, and we measure this phi change, and it's indirectly correlated to the presence of biomarkers of interest. So if you look at here, these are fiber optic interferometry, where an optical fiber can also be converted to an interferometric setup by means of creating some interfaces. So if you look at here, you have a core and surrounded by a clad. The dark blue is the core surrounded by a grey color clad, and when you allow light to travel through, we have created interfaces, first interface and then the second interface. So the distance between the first and second interface corresponds to the path length the light is being taken to travel through. And we, in the cladding, either you remove the cladding and introduce the biosample of interest and measure the output light at the other end, which would have created a phase change phi compared to the input light source. And in case of Maxender interferometer, you could see here I is input light, In is the input light, and I out is the output light. So you could see the changes in the phases. Here the input light represented by yellow color, and here you could see the green color changes in the phase of the output light. Why? Because on the distance between the first interface and second interface, where we have coated with some bioreceptors. So those thread-like structures represent the bioreceptors or the analyte which is being introduced here, due to which it created a phase change. Whereas in case of Michelson interferometer, we have created a two interface, and we measure the reflected light by keeping a mirror at the end of the optical fiber. At the distal end, you keep a reflecting surface so that the light will be reflected. So here this is the sensing region. At the end, distal end acts as the sensing region, and we collect the reflected light back, which shows the changes in phi. And in Fabry-Perot, as I mentioned already, it acts as a resonant cavity. So the interface, the distance between the first and second interface, it acts as a resonant cavity, and we measure the reflected light, which produces a changes in the phase compared to the incident light, with which we comment about the number of biomolecules present at the sensing region. Clear? So this is about the interferometric biosensors. And already I mentioned we are going to utilize nanoparticles for sensitivity enhancement, for improving the detection limits. to identify one molecule among the 10,000 molecules at ease without any difficulty and with 100% accuracy. We want to detect that. For that we are going to employ nanoparticles. So we'll see a bit of physics here, why these nanoparticles are employed, and the phenomena it is called as surface plasmon resonance. This is about a bulk nanomaterial. How the bulk gold will look like. Jewelry gold, you all would have seen that, right? How it looks like, bulk gold. Haven't you looked at a gold, any gold ornament like earrings, ring, bangle, how it looks like? I have given in the slide also. It appears yellow in color and it is lustrous and it's shiny. Yes or no? Yes or no? Are you all there?
Yes, ma'am.
Yes. But when I bring down the bulk gold metal to a nano sized material, so the same bulk gold by means of some mechanical etching or some chemical treatment, I am bringing the jewelry gold which is appear in the macro dimensions where I could see a lustrous shiny yellow color, and I am bringing it to 1 nanometer size, say for example. When the size is scaled down, you could see the nano sized gold appears red in color. This is called excellent optical properties exhibited by the nano gold when the size is reduced. So that exhibits enhanced optical absorbance. So what happens when I bring the bulk metal to a nano sized gold or nano metal? What happens? What is the physics behind? And we define a material to be nano if it is less than 100 nanometers. Okay, so we have taken a nano metal. When they are scaled down, the first change is there will be changes in the energy band gap of the nano metal. So if you look at the bulk metal, what will be the energy gap of the bulk metal? We know, right, we have a conduction band, valence band, and there is a band gap between the two, and accordingly we define materials to be three categories. So what will be the energy band gap in case of gold? Gold, it's a metal, right? That means it's a conductor. So for conductors the energy band gap is very less or zero band gap for metals or conductors, and a larger band gap for insulators, and a moderate band gap for semiconductors. When this bulk metal is transformed to a nano metal, there will be a band gap exhibited. You can see Eg nano is greater compared to the Eg bulk, due to which it exhibits excellent optical and electrical properties. I will tell you what are those optical and electrical properties. And when I bring a material, here I am telling you an example of gold. Whatever the material you take, when you reduce the size from the macro to nano, there will be high surface area to volume ratio. So if you look at a large square here which is of 1 meter size, the area is 1 to 6 meter square. So when I reduce the size of this large square into multiple smaller squares, and when I bring it down to nano, you just imagine when it is bring down to nano. So you could see here at the bottom where the area is 18 meter square. So when the macro is reduced to nano, the surface area to volume ratio is high, thereby you have larger surface area to work with. And when it is brought down to nano, the reactions are very quick. So the material become very active and it is very quick, and it's ready to involve in any kind of reactions like chemical reaction or whatever the reactions. That is what we are going to look at. So we are going to utilize these excellent properties of nanomaterials, and that's called as plasmon resonance. So if you look at a conductor, how do you define a material to be a conductor? Can you just brief the atomic structure of a conductor? which differentiates a metal to be conductor insulator depending on the atomic structure. Yes? Have you heard of the term valence electrons? What do you mean by valence electrons?
Electrons at the outermost shell.
Electrons at the outermost shell. So in case of conductors, how will it be?
There'll be free electrons.
There will be free electrons, or the valence electrons are higher so that it could be easily kicked out. That's why it's called a conductor, where it is ready to involve in for subsequent reactions with another material or with another component, right? So the surface plasmon resonance is when I bring gold. So here the plasmon resonance which I am talking, it's applicable only for the noble metals, gold and silver. Okay, even other metals exhibit this plasmon resonance, but they are not in the visible region. So we are particularly for biosensing applications, so we consider this noble metal, gold and silver, where they exhibit this plasmon resonance at the visible wavelength. Okay, I'll come back to what is the plasmon resonance. So as he pointed correctly, we have this gold metal and it's a conductor. It's having a higher valence electrons, and these valence electrons, when I irradiate light, when I incident light, and now it is in the size of a nano, right? So I have reduced the size of the gold to be of nano dimension, and I irradiate light. What will happen when the incident light intensity is in resonance with the collective oscillation of these valence electrons? So when they are at the nano regime, these valence electrons undergo collective oscillations or vibrations. When this vibration in resonance with the incident light wavelength, then that is called plasmon resonance. Plasmon is nothing but the collective oscillation of the valence electrons at the surface of a nanoparticle or a nanofilm. So if you look at here, I have given you a figure where you could see an inverted triangle, right, which is nothing but a prism. So from prism we observe dispersion, right? So we have taken a prism which acts as the dielectric metal, and on top of it I coated with a gold film of 50 nanometer thickness. So it's a nano layered thick gold. So since it is in nano, the valence electrons, that means the conduction electrons under the nano thick gold, will be undergoing oscillation. I incident light onto it. So due to the collective oscillation and the incident light wavelength, when they are in resonance, resonance is nothing but the frequency matching conditions, right? So when they both undergo resonance, there will be you could see at the other end you have a detector which collects the reflected light. There will be enhanced optical and electrical properties. So you could see here evanescent wave. So at the metal dielectric interface, we have this evanescent wave present, and the plasmon generation is represented by an exponentially decaying field. There will be enhanced oscillations when these film and the incident light are in resonance. So due to which you could see a particular light from the input spectrum is reflected. So what I will do, on top of the gold film, I coat it with antibodies or any bioreceptors, and when the analyte comes and binds, there will be refractive index changes happening at the surface of the gold film. Due to the surface refractive index changes happening at to the gold film, and you could see a particular angle of light being reflected out, and it is enhanced so that you could detect even one among the 10,000 molecules present in the biosample. And this is completely dependent on the nano dimensions, so the size of the nanomaterial. Here it is a thick layer, gold film. Since it's a film continuous, so the oscillations will be continuous, and that's why it's called a surface plasmon resonance. If you look at a nanoparticle, individual particle than a film, then it is called localized surface plasmon resonance, that is LSPR. Okay, here it is SPR because the film and the oscillation is continuous and it could propagate throughout the film structure. Okay, so it's completely dependent on the dielectric constant of the metal, that is the permittivity epsilon m and d, epsilon m and d of the metal and the dielectric material which you are considering, and you could see a particular wavelength being reflected. So from the particular wavelength reflected out, from its intensity or shift in the resonance angle, I can comment about the biomolecules present at the surface of the gold film. This is called surface plasmon resonance, and this particular configuration which I have showed you in the figure, the prism and the gold film, and then you have the analytes of interest or the biosample. This is called Kretschmann configuration, where we observe the changes in the resonant angle of the particular wavelength for detecting the biomolecules of interest. So same, instead of a film, if I have a nanoparticle, the golden colored particles represented here represent the nanosphere, nanorod, nanoshell, nanocage, star, crescent, and it's a plant virus being quoted, so AU plant virus. These nanoparticles can be synthesized or fabricated into different size, different shape. Accordingly, the resonant wavelengths will be varying. In case of this SPR, we observe the reflected wavelength. In case of LSPR, we observe the absorbance wavelength. So you could see here multiple peaks being represented, right? These are nothing but LSPR peaks. And if you have an anisotropic structure, then you have two LSPR peaks. One is the transverse LSPR and another one is the longitudinal LSPR. And these structures, they could be synthesized by using some wet chemical synthesis, or they could be generated by means of some physical fabrication techniques, like what to say, you can use some lithography techniques or some etching, so that you create gold structures at different shapes and geometry. So any of these techniques could be employed to fabricate these structures, and when these structures are fabricated, you see this excellent optical property of peak wavelength at a particular peak. Say for example, sphere, it exhibits 530 nanometer absorbance wavelength. So I know for a sphere of a particular diameter and size and shape, it has its peak absorbance at 530 nanometer. And then onto the surface of this nanoparticle, I coat it with the bioreceptors and then introduce the analyte. What will happen? The peak absorbance wavelength, that is the 530 nanometer, there will be a change in intensity or change in the wavelength shift because of the refractive index changes introduced by these biomolecules onto the nanoparticle surface. This is called LSPR. When the collective electron oscillation is localized around a single particle, then that is LSPR. If you have a film, then that is called surface plasmon resonance. And we are interested in measuring the extinction, which is nothing but absorbance plus scattering, which is dependent on R, particle size, and then the dielectric constant epsilon m and d of the metal, and then the dielectric material which you are choosing for developing the substrate. So if you look at the commercial pregnancy test strips, they employ these gold nanoparticles, and as I pointed already, the gold nanoparticle, they appear red in color. So that's why you could see onto the pregnancy test strip T and C line, a pink line will be measured, which is nothing but the nanoparticle which is conjugated with an antibody, and when the analyte comes and binds, they exhibit this response. Clear? Any doubts? Are you able to follow? Any doubts up to this? Yes or no? I keep repeating this, whether you understood something or not, whether you understood something or so. Please try to respond. So till this we have covered what a biosensor is, what are all the key components or the building blocks, and there are different categories depending on the different transactions. Here we are discussing only about the fiber optics, interferometry and surface plasma, which is given in your syllabus or the portions, I think third unit, fundamentals of optical biosensors. So we are looking at these categories. Under fiber optics we covered evanescent wave absorbance biosensors, fiber Bragg ratings. Evanescent wave absorbance biosensors, they are intensity modulating sensors where we are interested in measuring the changes in the intensity. FBG, fiber Bragg ratings, wavelength modulator, where we are interested in measuring the reflected wavelengths by fine tuning the grating interval. And we have discussed interferometry for measuring the changes in the phase. And for the sensitivity enhancement we have looked at SPR, where we are employing gold film of nanometer thick, and it's LSPR when they are localized into individual particles and they exhibit absorbance. Here they exhibit changes in the resonance at a particular wavelength of the reflected light. Okay. And another one thing, so this is the physics behind. So first is the SPR. A is the SPR sensor and B is the LSPR sensor. So A, the yellow colored thing, corresponds to the gold film coated onto the dielectric material, and when you introduce light, so we have a prism here which is a Kritzmann configuration. So when you introduce light, there is an evanescence being generated, and into the light blue cuboid which has been represented, where you can introduce the sample, you monitor or measure the reflectance. So the B, which corresponds to the LSPR, the yellow circle represents the gold nanoparticle, the collective oscillations due to which we have positive charges on one side, negative charges on the other side. So when they undergo this oscillation, you could see enhancement in the electromagnetic field, and when you irradiate light, it exhibits a peak absorbance. Clear? And as I mentioned already, the sigma extinction we are interested in. So here you could see in the figure nanosphere of different sizes. When you increase the size, eventually the color of the solution will also change, and in turn the peak absorbance wavelength will also be changing. They will be having a particular, at particular wavelength they will be having the peak absorbance, and it's dependent on the geometry of the particle and then the dielectric constant of the metal. Okay, so this is about SPR and LSPR. There is one more phenomena called surface enhanced Raman scattering. That's called SERS biosensors. What is the Raman scattering? What is Raman scattering? What is scattering generally? You incident light onto an object, and the light incident on the object will be scattered or reflected, will be reflected in all the directions. So Raman scattering is very particular because the Raman scattered light gives you information about the molecular composition of the material onto which you have shine light. So if you irradiate light, this is a classic example of a water molecule. So what are all the bonds involved in water molecule? So water molecule, if you take, it's H2O, right? So we have OH stretching. So if you irradiate laser, the chemical composition, which is nothing but the OH peaks or vibrational bonds, OH vibrational bonds could be identified by measuring the Raman scattered signal, which is called Raman spectroscopy. Onto the right side you can see a plot that individual numbers which I have represented, the wave numbers 912 centimeter inverse, 1479 centimeter inverse, this corresponds to the molecular composition of the material. But it's a weak phenomena. This Raman spectroscopy, it's a very weak phenomena, and if we could enhance this phenomena, then you could identify the chemical composition of the materials at ease. So how to enhance this Raman scattered signal? Again we are going to take the help of a nanomaterials. So you could see here two gold nanospheres. They are kept at a particular distance, and when the green color star represents the analyte of interest, I have kept these two nanospheres at a particular distance and I am irradiating the light onto these nanoparticles. when they are at a particular distance. This is called hotspot generation. So lambda by 10. So when they are kept at 2 nanometer distance, they generate hotspots, and the Raman scattered light from the hotspot is huge. So if you look here, when this green starred analyte is just kept onto the substrate, and when you monitor the Raman scattered signal, you see a black line, but it has the characteristic peaks, but you are unable to appreciate. And when they are kept onto a gold substrate, you could see some red where the peaks at near 1200 1400, they are not clearly visible. And when you place the star in a hot spot and then you collect the Raman scattered signal, you could see these peaks are prominent, and from these peak numbers you could comment about the chemical composition or the molecular vibrational bonds involved in the green star molecule. That is the principle of this surface enhanced Raman spectroscopy, where the vibrational bond structure of any molecule could be detected. So it could be employed in forensics, in archaeology to identify the metal composition of idols, and in clinical diagnosis to identify the pathogens. Again, the applications are not limited. So to achieve that, basically we have to trigger out this electromagnetic enhancement as well as chemical enhancement. So chemical enhancement is nothing but where due to the charge interactions it could produce a 10 raised to 3 fold improvement. In case of electromagnetic enhancement, which is from a nanoparticle, so you could enhance, the enhancement factor could be up to 10 raised to 11. So how to utilize this? We are going to look at graphene gold nano composites. So what is graphene? Have you heard of the term graphene?
It's a lightweight material.
It's a lightweight material. So we have sports cycles and other structures being fabricated utilizing these graphene materials, right? So basically it's a carbon material. So it is synthesized from the pencil lead which is available in a pencil, right, writing pencil. So the lead which is available in the writing pencil, which is in the bulk form, graphite, and I am bringing down the bulk graphite into a nanometer thick graphene. So when I scale down the material from bulk to nano, as I already pointed out, there will be enhanced optical properties, electrical properties. So when I have this nanometer thick layer of graphene, it has multiple functional groups active that could be utilized for bioreceptor immobilization, and it could produce chemical enhancement because of the multiple active functional groups present on the graphene surface. And due to the charge interactions you have chemical enhancement. And when I coat gold onto this nanometer thick graphene, so gold nanoparticles could produce electromagnetic enhancement, graphene could produce chemical enhancement. So I am tapping here the synergistic enhancement due to both graphene and gold. So with the graphene I could improve the bioreceptor density, and I could suppress the background fluorescence, and I also could extract better Raman signal. So you could see on the right side where it reduces the fluorescent background signals while acquiring Raman. So the red color, if you use a Raman signal, the individual characteristic peaks are prominent, and I could tap those. So this is one of the research study where we are studying this graphene gold nano composites. So this is how it looks like. You have the graphene nanometer thick layer represented by the hexagonal carbon structures intercalated with one another, and that could produce the chemical enhancement because of the charge interactions. And I introduce another nanoparticle, nanosphere, represented as green over here. And on top of it, when the analyte comes and binds, analyte is nothing but, we have taken a classic example, glucose. So when glucose comes and binds, if you have the bioreceptor coated already, you could appreciate the presence of glucose by monitoring these Raman peaks alone. So if I have a peak at 600 centimeter inverse of particular intensity, then I could comment about there is 1 mg per dl of glucose present in the sample, or 100 mg per dl glucose present in the sample. So that is the application of this graphene gold nanocomposites, where it could be utilized for improving the bioreceptor density and then the orientation of these biomolecules, and I could get a better signature. So in one of our research study where we employ this graphene gold nanocomposite, so this is the graphene from the pencil flakes. We have not done anything, we have taken that lead and we grind it into powder, and from that we have synthesized this graphene gold nanocomposites. Already as I mentioned, gold, it has a peak absorbance, so near 530, and also graphene, you could see the stem structure where this gold is spread onto the graphene layer, due to which what happens, there is an enhancement in the Raman scattered signal, and we have employed this for detecting pesticides in herbal products. So we have developed the 3D structure which I have represented here with a hexagonal carbon atom intercalated with each other, and on top of it we have the pink colored nanospheres. So when I irradiate with a laser and when I collect the Raman scattered signal, I could comment about the chemical composition of the herbal drugs or the herbal products. So with that we have collaborated with an industry to identify the contamination present in cardamom powder samples and in few of the greens where they face problem while exporting. At the export they have a quality check and they are unable to identify the presence of these adulterants with the conventional equipments also. So where this graphene gold nanocomposites could differentiate 0.1 and 0.6 ppm. So that means you try to appreciate the sensitivity enhancement which is being made possible with these nano materials which we are talking. Okay, so this is another study where the same graphene gold nanocomposite, now we are trying to utilize it for detecting micronutrients. So the deficiency of micronutrients in a blood sample could be monitored with the help of these graphene gold nanocomposites. So where we are developing an SCRS substrate by combining these graphene and gold together, and you irradiate with light and you will get to see the Raman scattered, enhanced Raman scattered signal. And with that here in this study we are trying to identify the concentration of zinc ions. So we are trying to identify the deficiency of zinc, vitamin D, iron. So if we could detect these micronutrient deficiency in an early stage, most of the diseases could be avoided. So that is the idea. So here we have used wet chemical synthesis for synthesizing these nanocomposites. So with that I'll stop over here. I have covered almost all the topics listed in your 1 credit course. Now open for questions. You can ask any questions if you have. Any questions?
Thank you, Dr. Gauri.
Thank you, Alvin. So if there are no questions, we can end the session.
Sure. Students, if you have any doubts, only mail to Dr. Gauri. She will respond. Thank you, madam.
Yes, thank you. Thanks, Alvin.